Hello and welcome to Bio40 Pre-Class Lecture number 4. Today we're going to continue talking about multifactorial inheritance. Last time we talked about what happens when two gene products can interact to form a novel phenotype. Today we will also talk about complementary gene action, epistasis, where one gene covers the phenotype of another, and finally redundancy, where multiple genes can serve the same function. These different types of interactions stem from how the gene products function in biochemical pathways. And each type of interaction will have a specific phenotypic ratio in a dihybrid cross. Here is a scenario where we see complementation. Once again, we have two genes, A and B. This time they function in a single pathway that will create a red pigment in flowers. A dominant functional copy of both genes is needed to make the red pigment. Let's look at the dihybrid cross again. 9/16 of the offspring will have at least one dominant allele for each gene and produce a red pigment. The other 7/16 will be homozygous recessive for A and or B and produce no pigment. This results in a phenotypic ratio of 9:7. We call A and B epistatic to each other. Either is capable of masking the phenotype of the other. If a flower is homozygous recessive for A, it will be white regardless of the genotype for B. Likewise, being homozygous for B makes A's genotype irrelevant. Complementation is interesting because it can allow two true-breeding individuals to have offspring different from either parent. Here's an example of a flower with pigment genes C and P. Again, dominant copies of both C and P are needed in order to make the purple pigment. We can call the purple flowers wild type, essentially meaning this is a dominant phenotype found in nature, while the abnormal white flowers lacking pigment are called mutants. We can take two true-breeding plants with the same white phenotype, one with a dominant C allele but no dominant P allele, and the other with a dominant P allele but not C, and when crossed, you see wild-type purple plants. The two parents complemented each other, each providing the allele the other lacked. When two parents with mutant phenotypes produce wild-type offspring, we say they are from different complementation groups, meaning they have mutations in different genes. This will be important to understand for a yeast lab later on. Now let's look at another situation. Again, a dominant allele of both A and B are required to get our final red pigment for our flower. But in this pathway, if A is functional but B is not, we can get an intermediate pigment which is blue. So let's bring up our dihybrid Punnett square again and look at our phenotypes. Once more, we see 9 times out of 16 there's at least one functional copy of A and B, so we get the final red pigment. In 3 cases, there's a functional A allele but no dominant B allele, so the intermediate blue pigment is formed. And in 4 cases, the flowers are homozygous recessive for A and no pigment is made. This results in a 9:3:4 phenotypic ratio. We call this recessive epistasis and say A is epistatic to B. If there is no functional A allele, the effect of B is hidden. The flower will be white whether there's a functional B allele or not. B does not mask A in the same way. Plants homozygous recessive for A are always different from homozygous dominant ones and heterozygotes. Don't be fooled by the order of the genes. Here's a slightly altered situation where B comes before A in the pathway. The Punnett square looks a little different, but you still see the 9:3:4 ratio. This is still recessive epistasis, but now it's B that's epistatic to A. Now here's a form of dominant epistasis. Again, there's a final red product and blue intermediate. But here, gene B functions to inhibit the production of the intermediate. So the intermediate, and therefore final product, only form when there is no dominant B allele present. Looking at the dihybrid Punnett square, there are 12 boxes with at least one functional B allele, which therefore make no pigment. Of the remaining 4 boxes that are homozygous recessive for B, 3 have a functional A allele and produce red pigment. The last box lacks the dominant A allele and will be stuck at the blue intermediate. This results in a 12:3:1 ratio. Here, we say the dominant B allele is epistatic to both the dominant and recessive A alleles. A dominant B allele masks the red phenotype from a dominant A allele and the blue phenotype of the recessive. Here's another form of dominant epistasis. In this pathway, the B gene again inhibits the production of the intermediate. But here, the intermediate doesn't have its own phenotype. So we bring up the Punnett square. Again, the 12 cases with the dominant B allele make no pigment, And even without a dominant B allele, there's no pigment made when the flower is homozygous recessive for A. Only the 3 cases that are homozygous recessive for B and with at least one dominant A allele will produce pigment. This means a phenotypic ratio of 13:3. Here, we say B is only epistatic to the dominant A allele. The presence of the dominant B allele doesn't mask the phenotype of a flower homozygous recessive for A because those phenotypes are the same. The last type of interaction we're going to talk about are redundant genes. Here, a functional dominant allele of either A or B is sufficient to make our pigment. So here's a dihybrid Punnett square again. 15 of the boxes have at least one dominant A or B allele and will make red pigment. Only the one box that is homozygous recessive for A and B will not make pigment. So we get a 15:1 phenotypic ratio. This table summarizes the types of interactions we've talked about today. Don't worry about remembering which type of dominant epistasis is 1 and which is 2. Just remember that there are 2 types and why they have different phenotypic ratios. Now let's talk about some complicating factors that stop us from being able to predict phenotype based on genotype. There's variable expressivity, that is, individuals with the same genotype may show differences in the intensity of the corresponding phenotype. Additionally, there's incomplete penetrance, which means that some individuals with a certain genotype just won't display the corresponding phenotype at all. Here's an example of fur color in tribbles to help us tell the difference between penetrance and expressivity. Let's say there's an allele that causes tribble fur color to be brown. When penetrance is complete and expressivity is constant, all individuals with the brown fur allele will have identical fur. When penetrance is incomplete, some individuals will just not have any pigment at all. Whereas, when expressivity is variable, all the individuals have brown pigment, but there's a range and degree of pigmentation. Some traits can even show both incomplete penetrance and variable expressivity. All these individuals have the same genotype, but the phenotype can vary wildly. In humans, we see variable expressivity in the disease neurofibromatosis type 1, an autosomal dominant disease of the nervous system. Heterozygotes can have extremely variable phenotypes. Mild cases manifest as small discolorations called café au lait spots, and a few small benign tumors called neurofibromas, while severe cases can involve thousands of neurofibromas, learning disabilities, and bone deformities. So what could cause genotype and phenotype to not neatly correspond? It ends up there are several possibilities. Environmental effects can change how genotypes manifest. Sometimes there are gene interactions we just don't know about yet, and some processes just happen to be governed by chance. Here's an example of how environment can affect phenotype. This example can even be observed within a single individual. This cat has the same genotype for brown fur all over its body, but the gene product responsible for making the pigment is temperature sensitive. It doesn't function well at higher temperatures, so the warmer parts of its body are light-colored, while the cooler, more distant parts are dark. Another example where genotypes can manifest differently is sexual dimorphism. Males and females could have the same genotype, but display different phenotypes. Male pattern baldness is not an X-linked trait. Both men and women can have the genotype for it, but it shows a different phenotype in men due to hormonal differences between men and women. This is called a sex-influence trait. There are also sex-limited traits that only appear in one sex because it affects a structure or process that's limited to one sex. For example, there's a fruit fly mutant called stuck that stops males from being able to separate from females after mating. That'll do it for this topic. Next time, we move away from thinking of genes as abstract concepts and start getting into their physical basis.